Good day, everyone. By the way, we are the group 9. So, our assigned topic is all about FRAT and FRAP. These techniques play a crucial role in studying molecular interactions, protein dynamics, and cellular processes. In this report, we will explore the principles, applications, and significance of FRAT and FRAP.
Good day ma'am, good day classmates. In this video, I am going to discuss my assigned topic which is FRET definition and its process. So first is what is FRET? FRET means Forster or fluorescence resonance energy transfer, which means it is a distance of dependent physical process by which energy is transferred non-radioactively from an excited molecular fluorophore which is the donor and also acceptor by means of intermolecular long-range dipole-dipole coupling. So, resonance energy transfer also can occur when the donor and acceptor molecules are less than 100 A of an another. So, energy transfer is non-radioactive, which by means the donor is not emitting a photon which is absorbed by the acceptor. Fluorescence or fluorescence resonance energy transfer can be also be spectrally shift the fluorescence emission of a molecular combination. Fluorescence microscopy coupled with the development of new fluorescent probes, which is FRIT, is a powerful technique used by the students for studying molecular interactions inside the cell with the improvised spatial, which angstrom, and temporal nanosecond resolution, distance range and sensitivity, and broader range of biological applications. FRIT can be accurate measurement of molecular proximity at last with a distance of 10 to 100 and highly efficient if the highly efficient distance is when if the donor and acceptor are positioned within the first radius is 3 to 6 typically 3 to 6 nanometer. So furthermore, fit is efficiency is dependent on the inverse sixth power of the intermolecular separation. Technological advances in light microscopy imaging combined with the availability of genetically encoded fluorescence proteins provide the tools necessary to obtain the spatial and temporal distribution of protein association inside the living cells. Process of FRIT or fluorescence resonant energy transfer. So in this slide, we assume resonant transfer is resonant energy transfer between the two molecules of fluorophore acceptor and the donors. In this slide, we assume that there is a two protein, which is the cyan fluorescence protein and the yellow fluorescent protein. CFP serves as the donor and the YFP serves as the acceptor. Fluorescent resonant energy transfer serves as or used to study protein-protein interaction. So in this slide, we assume that cyan fluorescent protein serves as the protein A and yellow fluorescent protein serves as the protein B. So the process starts with the excitement wavelength that gives energy into the cyan fluorescent protein, and then that energy will be donates into the yellow fluorescent protein. After that, the transfer energy will be occur since they are close to each other, which has the distance of 6 to 10 nanometer. But if the distance between the protein A and the protein B is more than 10 nanometer, there will be no interaction between those two protein, and the resonant transfer energy will no longer occur.
FRAT finds numerous applications across various scientific fields. Let's discuss some of them. So we have number 1, protein-protein interactions. Number 2, molecular proximity. Number 3, intracellular signaling. Number 4, DNA or RNA analysis. Number 5, drug discovery. Number 6, membrane studies. And number 7, nanotechnology. So first, we have protein-protein interactions. FRAP can be used to investigate interactions between proteins in living cells by attaching donor and acceptor fluorophores to specific proteins. Changes in FRAP signals can indicate protein-protein binding, protein conformational changes, or complex formation. So as an illustration for protein-protein interaction, we have here an example where the samples interact and show what happens. The first thing is that CFE gets excited, and what happens then, CFE as the donor will emit blue light. Since they are directly interacting and the fluorophores are very close by, we can measure a first energy transfer from CFE to YFE. And YFE as the acceptor in this case emits light. So if we see YFE emitting light, we have basically proof that the proteins are very close and are interacting. Another application of FRAT is for molecular proximity. FRAT can provide information about the distance and orientation between two biomolecules or subcellular structures. It helps in determining the proximity and arrangement of proteins, DNA, or membranes, aiding the study of molecular organization and structural dynamics. Next is for intracellular signaling. Breath-based biosensors enable the real-time monitoring of intracellular signaling events, such as calcium ions, pH changes, or second messenger molecules. By incorporating breath pairs into these biosensors, researchers can visualize cellular signaling processes under spatiotemporal dynamics. It also applies for DNA or RNA analysis. PRET can be utilized to study DNA or RNA hybridization, probe specific sequences, or analyze conformational changes. PRET-based assays are widely used in nucleic acid analysis techniques like DNA sequencing, genotyping, and detecting single nucleotide polymorphisms. In drug discovery, PrEP plays a central role in drug discovery and development. It helps assess ligand receptor interactions, screen compound libraries for potential drug candidates, and study drug target engagement in living cells or animal models. In terms of membrane studies, PrEP can shed light on membrane dynamics, lipid bilayer properties, and protein-lipid interactions. By employing PrEP approaches, researchers investigate membrane conformation, lipid-protein interactions, or membrane fusion events. And for nanotechnology, FRAT is utilized in nanoscale systems to prove molecular interactions, study energy transfer in quantum dots, develop biosensors, and monitor nanomaterial behavior. It aids in understanding and developing advanced materials with improved optical properties. These are just a few examples of the diverse applications of PrEP. Its versatility and ability to provide real-time, quantitative information make it a valuable tool in various scientific disciplines, be it in cell biology, chemistry, and biophysics. The principle of Foster resonance energy transfer of FRET is based on the non-radiative transfer of energy between two suitable fluorophores, which are the donor and acceptor, when they are in close proximity.
Let's define what is FRAP, and here is the definition of FRAP. FRAP stands for fluorescent recovery after photobleaching. It is a technique In cell biology, where a specific area of a cell is illuminated to bleach fluorescent molecules, and then the recovery of fluorescent is observed over time, providing insight into molecular movements and dynamic within living cells. These are the following process of FRAPs. Photobleaching is a specific region of the cell exposed to intense light to bleach fluorescent molecules, rendering non-fluorescence. Recovery: the cell is allowed to recover, and the movement of the fluorescent molecules into the bleach is monitored over time. Analysis: the rate and extent of fluorescent recovery are analyzed to understand the dynamic molecular movement and within the cell.
Fluorescence recovery after photobleaching, or known as FRAP, is a versatile and widely used experimental technique in cell biology. Its applications span various areas of research, providing insights into the dynamics and behavior of fluorescently labeled molecules within living cells. Some of the key applications of FRAP include the following. First, we have the protein dynamics and turnover, wherein FRAP is frequently used to study the dynamics and turnover rates of proteins within cells. It helps in understanding how proteins move, interact, and undergo turnover processes such as diffusion, binding, and dissociation from cellular structures. Membrane dynamics. FRAP is valuable in investigating the movement and behavior of molecules within cellular membranes. This includes studying lipid diffusion, membrane protein mobility, and membrane compartmentalization. Cytoskeleton dynamics, wherein researchers use FRAP to analyze the dynamics of cytoskeletal components like actin filaments, microtubules, and intermediate filaments. It helps in understanding their assembly, disassembly, and movement within the cell. So next, we have the nuclear dynamics. FRAP enables the study of nuclear proteins and their behavior within the nucleus. It provides insights into processes such as nucleocytoplasmic transport, chromatin remodeling, and the dynamics of nuclear bodies. Cell signaling and receptor dynamics. FRAP is applied to investigate the behavior and trafficking of receptors, signaling molecules, and other components involved in cellular signaling pathways. It helps in understanding the kinetics of receptor-ligand interactions and signaling complex formation. The next one, that is cellular transport and trafficking. FRAP helps in studying intracellular transport processes, including the movement of vesicles, organelles, and macromolecules within cells. Overall, the applications of FRAP are diverse, enabling researchers to gain valuable insights into the behavior and dynamics of fluorescently labeled molecules in live cells, in contributing significantly to various fields of cell biology and biophysics. For its principle, the principle of FRAP, or the fluorescence recovery after photobleaching, involves using a focused laser to irreversibly bleach a fluorescently labeled area within a cell. This creates a darkened region. Subsequent monitoring of the recovery of the fluorescence intensity in this area over time provides insights into the mobility, dynamics, and interactions of the labeled molecules within living cells.
Both Brett and Prap have revolutionized the field of fluorescence microscopy by enabling researchers to study dynamic processes and molecular interactions in living cells. These techniques provide quantitative and qualitative information about cellular processes that were previously inaccessible. By combining PRET and FRAP with other imaging techniques such as confocal microscopy or super-resolution microscopy, researchers can gain a deeper understanding of complex biological phenomena.